Hello students, I am teacher Svetlana and in our today's lecture we will learn about photorespiration, the C4 pathway also called as the H-Slake pathway and the CAM, that is your carcinosin acid metabolism, which is covered in the chapter photosynthesis. So let's begin. Photorespiration. Students, photorespiration occurs under the conditions like high temperature, bright light, high oxygen, and low CO2 concentrations. It is a wasteful process linked with C3 cycle, where instead of fixation of carbon dioxide, it is given out. That is, oxygen is utilized in place of carbon dioxide. It involves three organelles, that is chloroplast, peroxisome and mitochondria, and occurs in a series of cyclic reaction which is also called as PCO cycle. Have a look at this image here. Students, you can see here that the enzyme Rubisco acts as oxygenase at high concentrations of O2 and photorespiration begins when the RUBP, can you see here? The RUBP reacts with oxygen rather than CO2 to form your, so instead of carboxylation reaction, you have oxygenation reaction that is taking place, and it forms a 3-carbon compound, that is your PGA, phosphoglyceric acid, and it also forms a 2-carbon compound which is your phosphoglycolate. Later, it is converted into glycolate in the chloroplast itself. Then it is sent to the peroxisome. This glycolate is converted into glyoxylate by the enzyme oxidase, and it is converted into an amino acid glycine. This glycine is then further sent into the mitochondria. Now, in the mitochondria, glycine is converted into 2 molecules of serine and carbon dioxide is given out. You can see here carbon dioxide is released. Thus, there is loss of 25% of photosynthetically fixed carbon. Serine is transported back to the peroxisome and it is converted into glycerate. This glycerate is shuttled back into the chloroplast. Now here in the chloroplast, it undergoes phosphorylation and is utilized in the formation of the 3-phosphoglyceric acid, which gets utilized in the C3 pathway. Here you can see this, it is converted into PGA, where it is then utilized into the C3 pathway. So this entire process is called as photorespiration, where instead of carbon dioxide, oxygen is utilized, and the Rubisco, instead of undergoing carboxylation reaction, undergoes oxygenation reaction. The C4 pathway or the H-scale pathway. MDH and CR scale, while working on sugarcane, found 4 carbon compounds as the first stable product of photosynthesis. It's called as the dicarboxylic acid. It has been found to occur in tropical and subtropical grasses and some dicots. Some of the important plants are sugarcane, maize, sorghum, etc. The plants in which CO2 fixation takes place by Calvin cycle are called as C3 plants because the first product of CO2 fixation is a 3-carbon phosphoglyceric acid. So it's a 3-carbon compound, and hence it is called as the C3 cycle, and that's your Calvin cycle, and those plants are called as C3 plants. But in H-scale pathway, the first product of CO2 fixation is a 4-carbon compound, oxaloacetic acid, and hence such plants are called as C4 plants, and the cycle is called as the C4 cycle. Now, anatomy of leaves of the C4 plant is different from the leaves of C3 plants. C4 plants show Kahn's anatomy. Now, to understand what is Kahn's anatomy, have a look at this image here. In these leaves of such plants, the palisade tissue is absent. There is a bundle of sheath around the vascular bundle. Have a closer look at this image. This is your vascular bundle. Surrounding it, we have the bundle of sheath cells, and these are the mesophile cells around it. Now, the chloroplast in the bundle of sheath cells that are present here are large, but they are without or they have less developed grana, whereas the mesophile cells which are located here, they have chloroplasts that are small but they have a well developed grana. Have a look at this image here. It shows a comparison between the C3 plant and the C4 plant leaf anatomy. Here you can see these are the normal bundle of sheath cells. There is no chloroplast that is present here, and these are the mesophile cells which have the chloroplast, whereas in your C4 plants you have them arranged like this around the vascular bundle. This is your bundle of sheath cells. Surrounding them you have your mesophile cells, both having chloroplast, but here the chloroplast is not having grana, or it is having very less developed grana, and the mesophile cells have chloroplast which is small in size but they have well developed grana. Now students, let us see what happens in a C4 pathway. You have your atmospheric carbon dioxide. So the carbon dioxide is taken from the atmosphere and it is accepted by a 3-carbon compound here. This is your phosphoenol pyruvate PEP. In presence of the enzyme PEP carboxylase, it is converted into oxaloacetic acid, which is a 4-carbon compound, and this 4-carbon compound is immediately converted into malate in presence of malate dehydrogenase, and it is further converted into malic acid. Now this malic acid is transported to the chloroplast of your bundle of sheath cells. Hence, this malic acid, which is 4-carbon, is quickly converted into pyruvate, which is or pyruvic acid, which is a 3-carbon compound, and in the bargain the carbon dioxide is released. Now, since the concentration of carbon dioxide increases in the bundle of sheath cells, here you have the enzymes which are present for your Calvin cycle, that is the RUBP carboxylase. It participates in the Calvin cycle and it does not participate in photorespiration. As the concentration of CO2 is high, no photorespiration will take place, and we'll have carboxylation reaction taking place, and the sugar will be formed using the Calvin cycle, and it will be transported through the phloem. Now this pyruvate, which will be converted into pyruvic acid, is re-entered, or it is sent back to the mesophile cells, and it regenerates the phosphoenol pyruvic acid, or the phosphoenol pyruvate, again by the consumption of 1 ATP. Now, since the conversion results in the formation of AMP here and not ADP, can you see here? AMP is generated and not ADP, and hence for this 2 ATP molecules are required to regenerate ATP from the AMP. Thus, the C4 pathway needs additional 12 ATPs. So, in C3 pathway, we require a total of 18 ATPs for the synthesis of one glucose molecule, and here in case of C4 pathway, a total of 30 ATPs are required. But the C4 plants are better photosynthesizers as compared to your C3 plants, as there is no photorespiration taking place in these plants. Now students, we will see what is CAM, that is carcinosin acid metabolism. It is one more alternative pathway of carbon fixation found in desert plants. It was first reported in the family Carcinosaceae and so is called as CAM. Carcinosin acid metabolism. In CAM plants, stromata are scrotoactive, that is active during the night. So the initial carbon dioxide fixation occurs during the night time. Thus, the C4 pathway fixes CO2 at night and reduces carbon dioxide in the daytime via the C3 pathway by using NADPH formed during the day. So basically, during the night time, it carries on the C4 pathway, and during the daytime it carries out the C3 pathway. We have PEP carboxylase and rubiscose are present in the mesophile cells only. So there is no Kahn's anatomy present or shown by the leaves of these plants. See, have a look at this diagram here. You can see that during night time, when the stomata is open, carbon dioxide is taken in, and it is converted into the 4-carbon acid, that is your oxaloacetic acid, and it is then during the dark time itself, we have formation of malic acid, which is called as acidification or phase 1. And this malate is stored in the vacuoles during the night, and it is released during the day, which then it gives out the or releases the carbon dioxide during the daytime for the C3 pathway within the same cell, and this is called as deacidification phase 2. So here then, once the carbon dioxide is released, it undergoes the Calvin cycle, and the fixation of carbon dioxide takes place. So this is a chemical reaction where we have carbon dioxide fixation and its assimilation, where both are similar to that of the C4 plants. So this is called as your CAMP, or the carcinolycian acid metabolism. Example of CAMP plants are Opuntia, Aloe, etc. Student, have a look at this image here. For your quick and easy reference, I've put up a comparison between the 3 pathways of photosynthesis. The C3 pathway, or the C3 process of photosynthesis, which is your Calvin cycle. What basically happens is, to the stomata, you have the CO2 entering into the cell of your chloroplasts, where in the grana you have the rubisco, which takes it up and converts it into phosphoglyceric acid, which is a 3-carbon compound. So it's the first stable compound is your 3-carbon compound, hence it is also called as your C3 pathway, and the pathway proceeds to for the production of your glucose, sucrose, starch is ultimately produced. Whereas when you come to your C4 pathway, we have the Kahn's anatomy, that these leaves show, where you have this cycle stretched along the 2 different types of cells, that is the mesophile cells and the bundle of sheath cells. You can see here in the mesophile cells, when the atmospheric carbon dioxide is taken up, it is converted into a C4 product, that is your oxaloacetic acid, which is a 4-carbon compound, and then it is converted into your malic acid, and it is sent to the mesophile cells, where it gives out the carbon dioxide, and that carbon dioxide is taken up by your Calvin cycle, and the rest of the story remains the same as that of your C3 pathway. When you come to CAMP photosynthesis, here you do not have any Kahn's anatomy, but this cycle is completed during 2 time zones. That is, part of the reaction is completed during the night time, and the part of it is completed during the daytime. During the night time, you have your CO2 being taken up, converted into malate, and finally stored as malic acid during the night time. And during the daytime, this malate gives out the CO2, and when your stomatas are closed, and this carbon dioxide is then taken up by your Calvin cycle. So it shows a C4 pathway, as well as it is showing its C3 pathway also, so which is followed in this process of photosynthesis. So these are your 3 cycles of photosynthesis, which are used for the fixation of carbon dioxide. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.